So thank you everybody for connecting. I'd like to welcome Johannes Hohlbein. Thank you very much for making time to talk to us today. So this is the LIBREhub seminar series. We're talking about open source technology for bioimaging. But we talk about open source usually in quite a broad spectrum, open hardware especially, open instrumentation. And of course, open microscopy is an important focus in it. And the open microscopy movement has been a little bit parallel to the general open source hardware movement. And there, Johannes Hohlbein has been one of the leaders and one of the early people engaging in it and establishing resources. So it's a special pleasure to have him here with us today and sharing some of his insights into the movement and his projects. So thank you very much, Johannes.
Okay, thank you very much for the kind introduction. So I'm Johannes Hohlbein, and I would like to tell you something about open microscopy in the life sciences. Sharing is caring. So I'm located in Wageningen, somewhere in the middle of the Netherlands. And if you want to contact me, feel free to do that either on Blue Sky or via reaching out per email. So I would like to start with a microscopy image of a couple of gray dots placed on a gray background. And those are kind of Lactococcus lactis bacteria that are commonly used in the production of cheese. So what you see here is a bright field image using a relatively okay microscope, and basically boils down still that you see only gray on gray. So if we go back a bit into microscopy, then everything started pretty much with Antonie van Leeuwenhoek, who lived here in the Netherlands 250 to 300 years ago, and who was actually the inventor of the first microscope that was good enough to see and image single cells. Yeah, so the microscope you see here on the right, and if you look for it for the first time, you have no idea what's going on. And what it consists of is a metal plate over here. Then you see a tip where the sample was mounted on that small tip. And then you could move the tip in X and Y. You could change the distance of the sample to that small inlet over here. And you could change also the height of the sample. So you had very good control of positioning the sample. And now the most important thing is that inside here, between two metal plates, there was actually a lens incorporated that was then used to watch the sample or look at the sample from the other side and holding that entire microscope against the light to see something. Interestingly enough, he did not share any of the details on how he got the microscope to work. Yeah, so what was puzzling for most people for almost 100 years is how could he achieve the optical quality of the lens to be able to use this as a microscope. Yeah, so even at that time, there were compound microscopes were known, but due specifically chromatic aberrations, people did not really get a good resolution. Everything was kind of fringed out. And somehow, Antonie van Leeuwenhoek managed to have a really high optical quality that allowed him to look into the micrometer scale. Yeah, and it took basically another 100 years until the compound microscopes that we now know reached a quality comparable to that one shown over here. So these days, things are a lot easier, and open microscopy is plays a very important part. Yeah, so in the simplest case, you have a single lens that focuses onto a sample, and you can record an image with a camera. Yeah, so also people that presented probably already here, yeah, Vittorio and Benedict. So they developed, for example, a matchbox scope, yeah, which you can piece together for around $10, and which has a very decent resolution and allows you even to basically take pictures with a Wi-Fi enabled via your computer or via your mobile phone. Yeah, so $10 is what gets you into, as I still would call it, reasonably microscopy. So if you want to make that a bit more complicated, then you add another lens. Yeah, so use an objective and you project the image onto the camera. And there you have to spend a bit more. You can piece everything together. For example, with the open flexure microscope, that is, if you buy all the parts, do the 3D printing, you end up around $100, yeah, which will be good enough in all cases to image single cells and study already biology. And those are both of them very successful projects. So the thing is now we want to look a bit deeper. Yeah, so we'll take one cell and we want to zoom in a bit. And there was a very nice drawing. So this is the inside of an E. coli bacterial cell, yeah, made by Gutzel. And what we see here is a lot of many different proteins that are, for example, interacting with the DNA here shown in yellow, DNA interacting proteins, different proteins in between. We have a cell wall, we have a motor protein that can get the cell moving and so on. Yeah, all this structural information we got with X-ray crystallography, but in the microscopy world, it's much more difficult to really see single molecules and resolve them. So I'm going to focus a bit on this small molecule shown in the left corner. So this is a CRISPR-Cas multimeric protein. So in this case, type 1e cascade. And the big breakthrough for fluorescence microscopy came with the invention or discovery of the fluorescent protein. Yeah, so because this is a genetically encoded fluorophore that you can tag to your gene of interest, and you can now excite that fluorescent protein and measure its emission. And even though the protein is in the scale of the fluorescent protein, in the scale of the protein that you would like to study, so say roughly 10 nanometer, the issue is that fluorescence microscopy is diffraction limited. So instead of seeing the protein, you see a spot that is a lot larger than the protein itself. And the question is, how can we now get back from that large red blob into a region that is small enough to tell us something about the protein or its movement? So this is where single molecule localization microscopy comes into play. So we have our protein that is tagged with a fluorescent protein. Then we use a microscope. Again, we make it a bit more complicated. So in addition to the objective, lens and camera that I showed you before, we now couple laser light into the beam, yeah, so which is focused into the objective, gives us a parallel laser beam here, so that we can effectively, in a relatively small volume, excite that fluorescent protein and measure the fluorescence of that protein with the camera. So then on our camera, we have, I don't know, a megapixel or something, then every single protein will end up as a distribution of intensity values across a pixel. Yeah, so here I cut out from the camera with having, say, 1 million pixel, very small region presenting one exact molecule. Yeah, every pixel here represents 100 nanometers, so in total 500 by 500 nanometer. That's what we call the diffraction limit. And now we can determine the center of mass, or we can fit the intensity distribution with a Gaussian to kind of localize that emitter with a precision, say, in the order of 20 nanometers. So that's all good. And now we're getting into the realm of super resolution microscopy. Yeah, so we know that our intensity distribution of the fluorophore is always a couple of 100 nanometer. But we also know that if we fit the emission of a single molecule, we can get a localization precision in the order of 20 nanometer. So if we now have a structure where we have many emitters placed, then normally they would all overlap, and we cannot resolve any structure that is smaller than this value of roughly 250 nanometers. So the conceptual idea for super resolution microscopy was then to make our fluorophores present blink. Yeah, so if we have them in an off state and we find conditions where only individual emitters are fluorescent at any given time, we can localize them and can start over time reconstructing our underlying structure or the movement of a single molecule. Yeah, so meaning we can get from that cloud as a function of time over the localized single emitters. And the idea there is a bit that we separate the emitters in time to be able to separate them in space. Okay, so now we have the conceptual idea of the super resolution microscopy, and now we have to go into the practical realization of that. So what do we need? We need a microscopy hardware, we need software to analyze our data, and we need suitable assays. And the challenges that we faced and that everyone else is facing, so that you might not have all the expertise, you might miss the funds, and you might miss the time, the money, or sometimes in the worst case, both. So one inspiration for me, this is now kind of 6, 7, 8 years ago, was the open source microscope. That was kind of the first idea where people thought, okay, how can we simplify that? How can we make a microscope that is good enough to do the single molecule localization, but which is also reliable and can be basically built together from available components? And I looked at the microscope and I said, okay, it's very nice, but a few things I didn't like. So I've started designing basically our implementation of that, which we called the MyCube. And the idea was to design a single microscope body, so milled from aluminium that you can make done in the workshop. Then you can mount commercially available Thorlabs components that can hold filters and mirrors and so on. Complement the setup with a couple of 3D printable parts that hold, for example, lenses or additional lenses. And then basically what we have is we have an excitation path with a laser light coming in here. We can move this, translate the laser beam on that stage, focus the laser light into the objective. On the objective, we have a motorized stage to scan our sample. Fluorescence is detected then from here and passes through the dichroic mirror and is sent with a mirror towards the camera. So we made all of those plans available so that people can start building their own microscope available. Okay. And with that, kind of a proliferation of the MyCube started, because similar to what I did with the previous project, people thought, that's quite nice. Let's take a couple of parts, but I would like to have certain things different. So even from the MyCube alone, then a couple of new microscopes got spun out over the last couple of years. So for example, the MyEye from the Tutkus lab, they used kind of an elongated block here to have another layer for the excitation and detection. So then there was one implementation from the Gansinger group here in the Netherlands. They also made it a bit wider because they didn't want to have an XY scanning stage, but they wanted only have a very small piezo stage. Then the Cruz-Meyer lab saw the K2 turf, said, we want to make that everything a bit narrower. So they designed their thing. And then also a very detailed protocol came up from John Daniel and the Klennerman lab, who also made really hundreds of pages of that single build available. Interestingly enough, also this idea of the open source microscopy for high-end applications was then taken over by the French lab, and now with the open frame system there is a semi-commercial part available where people then can just buy parts and piece together their microscope. So again, their main invention and really cool thing is that they kind of came up with a cylinder that's stackable, so you can basically extend that depending on how many optical layers you want to add. Very nice system. Brings us to the question, proliferating standards. So I always like to show that XKCD comic. So situation, we have 14 competing standards. Someone comes and says, oh, well, that's completely ridiculous. We should start one standard that combines it everything, and that we can get rid of all those implementations. And then soon we're going to have the implementation with 15 competing standards. So in a way, I don't see that as a problem because choice is technically good for everyone. Okay, so with all those things in mind, I and Kirti Prakash, we kind of came up with a comment article published in Nature Methods to think about that a bit further. Yeah. So what do we have? What do we need? How can we support people? What might be missing and what might be avenues for the future? So I highly encourage you, if you're interested in open microscopy, have a read here. And in our definition that we came up with is that we see open microscopy as a movement to make scientific research involving microscopy, any associated data, and dissemination thereof accessible to all levels of an inquiring society. And we defined it relatively broadly. So we said, okay, we need, as mentioned earlier, we need the hardware, we need assays, and we also need software. And together with the paper, we started a small GitHub page where we try collecting some of the tools that are available, so for people to have one repertoire where they can look and search for things that have already been published in terms of hardware and microscopy. So I also highly encourage you, if you have a project that you know of that is not there, send me an email with a request to add it so that we kind of keep the library going. Okay, so what further did we do? Yeah, so this is kind of an antibody labeled microtubule network on a 7 cell, and here is the idea of super resolution microscopy put in action. Yeah, so normally we have a dense labeling of our structure, but with adding the right buffer chemicals, we make all those fluorophores blink, and we can then use that to reconstruct the image, get a higher resolution in 3 dimensions, so not only in X and Y direction, but also with playing some tricks here also in the Z direction. So over the last couple of years, we worked on a number of implementations, how to do the localization faster, how to do single particle tracking, how to do spectral multiplexing, worked on adaptive optics, correlative light electron microscopy, and I'll pick out number 2 and 3 just to show you, give you a bit of data how it looks like. So going back to our CRISPR-Cas molecule, put a fluorophore on it, and then we ended up with movies taken on a different microscope where we see the outline of different cells next to each other in green, and then we can track the movement of a single protein as a function of time. Yeah, so this is kind of a very long track that we got, relatively long for fluorescent protein. Very happy with that. And then we used the analysis of this dynamics to get an idea about what is this CRISPR-Cas molecule doing in a cell, how fast can it find certain sites on DNA, and so on. The other example I want to show is about spectral multiplexing. Yeah, so you often have the case that you would like to label different things in a cell with different colors. And because fluorescence emission is normally relatively broad, you quickly run into the condition that much of the spectrum basically overlaps. And remember, for our detection, because we need the sensitivity, we cannot detect colored light, but we always use a black and white sensor that only records several levels of intensity. So it's very difficult to change here to distinguish, for example, of emission of emitter in different colors. So what did we do? We came up with an idea where we took a relatively cheap transmission grating, and instead of putting that very far away from the camera, we put it as closely as we possibly could in front of the camera directly. So what will happen is that normally we see just a single dot, that's the zeroth order represented. But by using this grating, we generate a plus first order of the refracted light, which is then the distance between the zeroth order and the first order is depending on the wavelength of the light. So by basically measuring the distance between those two spots, we get an idea about the median emission of our fluorophore. Yeah, so on the right, you see a raw image. Every single molecule is now showing up in a pair, and we simply detect the pairs measure the distances, and then we know which of the floors we can have, so whether it's a short distance or a larger distance, and we can map that then back onto the image, including the super resolved position. So what do we need for that? So we have, say you have our single molecule localization microscope available. All you need is a diffraction grating, roughly $124 at that time. We need a couple of plugins, so an optional plugin to do some filtering in time, available in ImageJ, and then we released the plugin to do the fast localization. So we have an implementation on the Thunderstorm available, and we used created a different program which then finds those pairs and is able to map the distance between the localization spec. So all of the software is available on our GitHub page. So if you want to play around with it, feel free to go. If you do that, so to give you an idea on, so here we show the swarm image, super resolved. We labeled the microtubule in one color. We labeled the clathrine labeled pits in a different color, and now we don't only have the X and Y higher resolution, but we can also have with encoding the emission color in the color scheme, directly see where the different things are located. So with that, already coming to an end, I want to give you some final thoughts on open microscopy. And I think personally it is something really worth fighting for. So because still in the past and in many different labs, it's okay, we make everything available and you use data to publish a paper, but we have no idea of checking that. So how was it done? What data was recorded? How was it then the data analyzed? And in some labs, there's still very little attention to that. So you get flashy, shiny papers where people basically have no idea what's happened in the background, also severely limiting the reproducibility. So again, for you as a PI, for you as a student, if you care about that, you can make the change happen. Next point is time versus money. So in many instances, you have a lot of time in your hands. You can develop certain things for your microscopy. Sometimes as a PI, you have a bit of money, and I rather spend the money to buy something than spending my PhD candidate's time to develop something again, which was already developed. So brings me to the next thing. So this is why I personally think it's very good to have routes of commercialization, even in open microscopy. So because there should still be a pathway that by making things available, even selling small components, that you have some revenue stream that if you set up a company that you could benefit from that. As pointed out already several times during my presentation, I think hardware, software, and assays, they need to go hand in hand, especially for complicated things like single molecule localization microscopy. Without having assays, hardware, and software available at the same time, it's not of much use. And also for the open microscopy, I think it really is best done if there is long-term commitment. So that could be either on time or on development, because again, there are many different projects that are spun out quickly, but it's a lot more difficult to really keep them alive. And with that, I'm coming to my final slide. So I hope I convinced you that open microscopy can help to improve the accessibility of high-performance microscopy. And I showed you some of the things that we did, specifically the MyCube and the spectrally resolved single molecule localization microscopy. Would like to thank the people who did the work, Martijn for the spectrally resolved imaging, Koen and Sander for the developing of the MyCube and other members of the lab. And really most important point, your contribution matters, and we can do that only with everyone contributing. I'd like to shout out to the funding agencies and you for your attention. Thank you.
Thank you very much, Johannes. This was a fantastic talk.
Thank you.